structures prediction with AlphaFold. So, yeah.
Thank you everyone for coming. Today I'll be presenting on the paper that made AlphaFold 2. Paper by John Jumper and al. And it's published in Nature, July 15, 2021. Today I'll first be talking about what the problem of protein folding is, how AlphaFold 2 presents a viable and effective solution, and what are the results of AlphaFold. So why does protein structure matter? Well, after the Human Genome Project, scientists discovered that they figured out the entire human genome. So they know the amino acid sequence of every protein. However, although we know the amino acid sequence, we don't know the 3D structure of each protein. And this 3D structure controls the function. For example, for enzymes, the 3D structure controls where the binding pocket is. So what are some biological experiments or bottlenecks that happen when trying to determine 3D structure? Well, the three major ways of determining 3D structure via biology are X-ray crystallography, cryo-EM, and nuclear magnetic resonance. And all three of these solutions take months to years and are extremely costly. Likewise, simulating protein folding using direct molecular dynamics is inefficient as well. Trying to simulate using physics laws runs into a barrier where there are just too many atoms. For example, even small proteins with upwards of 30 amino acids have a hard time simulating folding, because you not only have to simulate the interactions between each atom in those 30 amino acids, but also the water atoms around the protein. So how is AlphaFold such an effective solution to this? Well, first I want to talk about AlphaFold's model at a glance. We have three main parts of the model. The first is input and pre-processing. So how does AlphaFold 2 store its data? How does it manage that data? The second is evoformer, or the encoder. Evoformer is basically the brain of AlphaFold. It figures out how to store the physical and structural data and how to use that knowledge to understand the protein structure. And the third is the structural module, or the generator. This part is the one that predicts the 3D protein structure. So how do we pre-process the data? This is a section of the model highlighted in red. Well, first we're given the amino acid sequence from the Human Genome Project. But the amino acid sequence is not enough alone just to predict the protein structure. We also need other types of data. So we also do a genetic database search to try and find evolutionary data for our multiple sequence alignments, MSA. What does this mean? Well, let's say we're trying to find out the 3D protein structure of hemoglobin in humans. AlphaFold is going to try to do a genetic search to find hemoglobin in species similar to humans, such as apes or monkeys. Next, we also try to find similar structural data. For example, if there are proteins already similar to hemoglobin in the amino acid sequence, and we know the 3D structure of that protein, we're going to try to find a similar 3D structure, which is called a template. Even if there don't exist any good templates, it's still useful for AlphaFold 2 to get in protein structures that look similar to its target protein. Finally, we try to combine structural and evolutionary features to make a pairwise matrix. This pairwise matrix compares certain features of amino acids. For example, one feature is amino acid distance. So how far two amino acids are located from each other in the chain. So amino acid 1 has a distance of amino acid 4. Has a distance of 3 from amino acid 4. So now we're getting onto the second part of the model, evoformer. As I said earlier, this is the brain of the model. It's the biggest portion of the model with 48 blocks. And it's also what processes the physical and evolutionary data to try and figure out the 3D structure. It uses attention, which is a mechanism in transformers, similar to how ChatGPT works, to try and understand how different amino acids affect each other. So the first and one of the most innovative methods is trying to figure out how biological genes throughout time have shifted mutations, and thus you can try to figure out a 3D structure. For example, these 4 columns highlighted in green, A, K, C, E, they seem to be conserved across all these species. This indicates that these 4 amino acids are very important to the structure of the protein. This is called conservation. However, over here you can see in these 2 red columns, a simultaneous mutation happens at D and C. All of the other amino acids show G and L. This indicates that because these 2 specific amino acids mutated in the same place in one species, it means they likely have a correlation with each other. This correlation implies that these 2 amino acids are close to each other in the 3D protein structure. So although they're not right next to each other in the chain, we now know a bit more biological or 3D structural information. The second main pillar of information that we try to use is pairwise physical encodings. So as you can see in the pair representation in blue over there, we store a pairwise matrix comparing each amino acid to another amino acid. AlphaFold's model learns how to represent the physical structure. For example, the distance between these amino acids in 3D space, the relative angle between them, and also other properties such as maybe how much electrical bond they have. One thing that made AlphaFold 2 so innovative though, is that this biological information and physical information didn't happen in isolation. Instead, as AlphaFold 2 is getting trained, the model communicates between these 2 pillars and tries to learn based off of certain biological information how to update the physical information, and based off of the physical information how to update the biological MSA sequence. Finally, we arrive at the third part of our model, the structure module. The structural module is in charge of predicting the 3D structure of the protein. So how does it do it? First, all the amino acids start at the origin of the 3D plane. Then, we try to predict the position and angle of each amino acid relative to the origin. So, we try to predict the position and rotation only with these 3 atoms as the backbone, the NCC. This way, we can worry about the side chains later. So, after AlphaFold 2 predicts the backbone of the protein in 3D space, it tries to predict at which angle is the side chain, notated here R, located. So one cool thing and one major innovation that AlphaFold 2 did was that it de-emphasized the sequential order. What does this mean? Well, we know that in a protein, amino acid 2 has to come after amino acid 1, amino acid 3 has to come after amino acid 2. However, AlphaFold 2 tries not to directly encode that into the model. Instead, it focuses on predicting the 3D location of each amino acid, and then at the very end, figuring out whether the entire protein chain is physically possible. This can lead to wacky results, such as mid-fold AlphaFolding producing impossible structures. For example, you can see the red protein chain over here connecting all the way to the red part on the other side of the protein. So we're done with the model now. So I want to talk about which features of the model increase performance and which ones decrease performance. So it turns out the authors wanted to do a study to see which parts of their model increase the most performance, and it turns out every part did. So for example, you can see this red box highlighting the baseline in black indicates training the model with all of the features attached. When we get rid of all these features, performance decreases to the left of the baseline. On the x-axis, you can also see there are 2 metrics that will also be important in later figures. GDT, which is a measure from 0 to 100, and measures how different the two proteins are on a global level. An IDDTCI, or interface distance difference test. And this measures how different the protein backbone is from the actual protein backbone. So how different is that NCC backbone from the ground truth? AlphaFold 2 is also open source, which means you can run it on your own computer. Here's the result of one AlphaFold 2 prediction. On the top, you can see we have this color-coded bars of PIDDT. PIDDT stands for Predicted Interface Distance Test. And the parts of the protein that are highlighted in dark blue or light blue means that AlphaFold 2 is very confident about the position of these amino acids. However, the parts of the protein measured in yellow and red means it's less confident. So there are two other novel architectural aspects that improve AlphaFold's architecture. The first is recycling, which is introduced by AlphaFold. What is recycling? So you might have seen that at the bottom of this figure, highlighted in blue, there's a bar that says recycling. What this means is that after we put in data into the evoformer and then goes through the structure module, that data is recycled back to the start evoformer and goes through the model 2 more times. This serves 2 purposes. Number one, it makes the model deeper and gives it more time to process information. And number 2, recycling the data allows the model to make sure that's consistent with its earlier predictions. The second improved method AlphaFold uses, this one is not introduced by AlphaFold, is noisy student distillation. It sounds really complicated, but in reality it's pretty simple. So the problem that AlphaFold 2 was dealing with was that there was just too little human-generated data at the time, only 150,000 protein structures. And this little protein structures didn't give enough training data for AlphaFold 2 to make good predictions. So what did they do? They created artificial data. AlphaFold 2 took in unlabeled amino acids and passed it into its half-trained model. And then it made predictions of the 3D protein structure. Predictions that had high confidence were considered as labeled 3D data. Well, it's fake because it's not experimentally determined, but it's labeled. And this labeled artificial data is passed back into the model to give the model more training data. The important part to note here is that although our labeled 3D data might be noisier and less accurate, it's still better to have more data rather than less. So now let's talk about results of how effective AlphaFold actually is. So the CAS competition is the worldwide competition to determine how effective models are on protein folding. It's been around since 2006, and they basically separate a few experimentally determined proteins that haven't been released to the public, and then let models test their accuracy on it. The metrics CAS competition uses the GDT, which is the global distance. As I said before, GDT measures how different the entire protein structure is from the real experimentally determined one. So you can see that in 2018, AlphaFold 1 increased performance on CAS by around 20%. And then in 2020, AlphaFold 2 increased performance almost up to 90. And a performance of 90 or above is considered almost the same as experimentally determined structures, indicating that AlphaFold 2 almost ends up to have performance as strong as the lab. So what made AlphaFold 2 so innovative? The first is that AlphaFold 2 had an end-to-end structure. You can see that from the input data to the 3D prediction, it's one straightforward model. We take in the amino acids on one side, the amino acid structure, the amino acid sequence on one side, and then on the other end of the model, we predict 3D structure with no breaks in between. Other previous models tried to have separate parts, maybe one for predicting the 3D structure, and then 2 before that for computing biological information, and then physical information. But these sections of the models weren't connected. For example, in AlphaFold 1. The second aspect that AlphaFold 2 surpassed in other models was focusing on deep learning, or basically a really deep AI model, instead of trying to encode too much physical or biological information. So you can see that when I talked about AlphaFold 2 today, there wasn't that much information on like physical laws or like chemistry. Instead, it just focused on building a really, really deep model, especially with the 2 other iterations added by recycling. This shows that at its core, when we use deep learning to learn representations better than if humans try to encode the physical laws itself. The third is that AlphaFold 2 switched to a new, more powerful architecture. AlphaFold 2 switched from convolutional neural networks, which are commonly used in image processing, to evoformer, which is a modification of transformers, the same type of architecture used in ChatGPT. What's the real world impact of AlphaFold 2? Well, before AlphaFold 2, at the time, there were only 150,000 protein structures. But afterwards, AlphaFold 2 has predicted 200 million proteins. A jump from 150,000 to 200 million. It's insane. This shows that AlphaFold 2 has predicted almost all known proteins. And this means that labs can find out the protein structure from years, and now it's down to minutes using GPUs. Also impacts drug discovery because it makes it easier to find out how the protein structure is. It lets us understand mutations better because we can simulate 3D structures at a much cheaper level. And it increases equity for small labs because now tiny labs don't need to buy expensive equipment and run long lab procedures to figure out protein structure. And for this achievement, the first author and CEO of Google DeepMind received the Nobel-Prize in chemistry, which just shows that how prevalent AI is becoming in mainstream scientific research. So to summarize, AlphaFold 2 solved a 50-year-old protein folding problem and won the Nobel-Prize. It combines evolutionary information, physical information, and deep learning to make a really strong end-to-end model, which means a model that takes an input out one side and doesn't stop until it predicts the 3D structure. And the cool part is that AlphaFold 2 is completely open source. All its weights are online, all its code is online, so you can run it yourself. Thank you.
So the next presentation is
I have a
Angelina.
question.
Yeah.
If there aren't, I was waiting for students, but if you guys don't have questions, I do. All right, so I have 2 questions for you. One is, as a X-ray crystallographer by training, do you think that this is going to just be the end of experimentally determined protein structures? I mean, they're approaching what we would consider good enough to be experimentally determined, right? So, what do you think?
Yeah, sorry, Dr. D'Souza, I think we only have time for one question, but personally, I don't know much about X-ray crystallography, but I think that since AlphaFold 2 is so powerful and it takes so much less time, so much cheaper to run, I think it will largely replace X-ray crystallography in the future.
I think so too.
So the next presentation is Angelina, and also we'd love to hear your feedback, so we'll be sending out feedback for you on the presentation.